Let's we move towards getting started. I want to welcome you to the our monthly Distinguished Lecture Series, and today we have a special treat because one of our new faculty, not really new anymore, but still new, Kathy Lord, will introduce our speaker today. So it's great to introduce Kathy Lord. Thanks everybody for coming.
So today our speaker is Jeremy Veenster Vanderveel, and Jeremy is the Ruane Professor of Psychiatry and the Division Head of Child and Adolescent Psychiatry at Columbia, and also the New York State Psychiatric Institute. I've known Jeremy, I think I met him actually when he was a high school student. He is in Chicago, and he was a Harvard undergraduate, won all kinds of awards. Came to University of Chicago for medical school and residency, and then also spent some time at Vanderbilt, and then Columbia was lucky enough to persuade him to come. He's also the interim director of the Center for Autism and Developing Brain, which is where I worked before I came here, so you guys be nice to him. Jeremy is a molecular and translational neuroscientist who does mouse work looking for new ways to treat disorders like autism and pediatric OCD. He is an associate editor for the Autism Research Journal. He is an editor for JAMA Psychiatry and has won all kinds of awards. I can also say from personal experience that, I mean, he's the real thing, you guys. He is a real scholar, scientist, researcher. He is also a wonderful clinician who is committed to doing good work and using what he's learning from research in that context. And he's a leader who just steps up in all kinds of sometimes interesting, sometimes awful situations. And I think, you know, that's what we all want to be. So welcome him. And today he's going to talk about pathways to new treatments for ASD. So we're really glad you're here.
Thank you. That might have been the nicest introduction I've ever gotten. So thank you. I'll try to compose myself a minute. So I looked like a high school student. I was not a high school student when Kathy first met me. I was, I guess, between my second and third year in college. I always looked young. Now that my hair is starting to go, I don't look quite as young anymore. But yeah, the first time I actually saw an evaluation of a kid who did have autism spectrum disorder, but not strict Connor autism, that first kid, it was with Kathy, and I was sort of a fly on the wall, and I just thought this is how you did it. And this was back in 94 or so, and it was the way Kathy did it, and now it's the way that good people across the world do it, but it was not at that time the way most people did it, which was really a treat. And that time at the University of Chicago was sort of magical time, experiencing folks who were thinking about some of these problems from all different angles, but we're very much a community, and I'm projecting on this group here that you're like that. So. And then one more thing before I start, which is to say, take good care of Kathy. We miss her a lot. I have the sense that she'll continue to make a huge impact here, but we were grateful for the time that she was in New York. I was grateful for the time when you were in Chicago as well, but know that you've got somebody really wonderful. So this is sort of a conceptual talk. There are pieces of it that are sort of right at the leading edge, but it is more conceptual about the way I think about treatment development. And I have some hidden slides that are more oriented toward mouse work. So if people ask me questions afterward, I can unhide some of those, but figured I would be more clinically oriented. It's important to acknowledge that I have had some funding from pharmaceutical companies. None of that has yielded a treatment that's available. And when that pops up, those will be highlighted in red. I should say it would be nicer if those were more robust disclosures to make, if we actually had treatments that had come out of those studies. And then, and this is not fully up to date, Julia's not on there, but yeah, in any case, it's about a year old. In any case, this is also to acknowledge that some of what I'll present is from my research group, some of it is from other groups as well, and that will be highlighted on the bottom, and I'll try to call that out. So because this is an autism group, I'm not going to spend a lot of time talking about what autism is. That feels a little silly. But I'll talk about how that challenges us, talk very briefly about the treatments that we've tested to date and how they've been tested with an eye towards some of the challenges moving forward. And then we'll talk about a couple of examples from what are, I would say, the two pathways that we can think about to treatment development. One is the molecular pathway, which now these days we call precision medicine or various other catchphrases, taking things from the level of understanding a risk factor that robustly increases the chance that somebody will have autism, trying to carry that forward in a more or less linear way to lead us toward a treatment that's actually targeted based on pathophysiology of autism. And the other is what we've by and large done in psychiatry, which is that we have a medicine that does something and we figure out maybe what it does something for. And then talk toward the end of the session about the idea that ways to tie together larger groups of individuals so that we don't have something that's targeted to something like Timothy syndrome, where you can have a very nice idea of a treatment that might be beneficial, but there are something like 40 or 50 people with Timothy syndrome and autism. You're probably not going to be able to at least conduct the traditional style randomized controlled trials that we think about in treatment development. So how do we tie larger groups of people together, ideally using biomarkers? And then come back in the end to talking about the reality, which is that we're not going to have treatments that are going to be by themselves biological, or I don't think so, biological cures, but that we're much more likely to have treatments that we hook together with behavioral interventions that perhaps allow people to benefit to a much larger degree than would otherwise be possible. So this is a quote I like from Isabel Rappin, who used to come and give the sort of bio side of the biopsychosocial introduction to autism at the Cold Spring Harbor course on autism that Dan launched and recently tried to back away from, at least in part, and leaving it to a few of the others of us. But autism is not something that is a disease in the way that we usually think about diseases in medicine. It's certainly not something that we're ever expecting to be a single disease. The way we define autism, we imagine that the symptoms that we use for diagnosis, which is really the only way to assess whether somebody has autism spectrum disorder or not. But those symptoms are likely the common final outcome of many, many different ways to get there. I think importantly, if you think about this diagram, you could also, depending on your perspective or depending on the child in front of you, put something else in the center. So for some kids, you might put their language difficulties or their intellectual disability in the center, and their autism in some ways may not be the most impairing thing that they're dealing with. But importantly, and this is one of Kathy, I don't know why my pointer isn't visible on my screen, but there you go. But these two pieces, in some ways, when autism was first described, ended up getting kind of lumped in, particular language impairment. Now we think about them as separate, and we think about them as separate targets for treatment. When somebody comes to see me as a psychiatrist, usually they're coming with these sorts of behavioral concerns around the edges, which we can often do something about. Medical comorbidities, however, should really be the first thing that we're thinking about when a kid comes in, and we'll come back to that to some degree, but I would especially note that if somebody has a seizure disorder, that should be what you're thinking about in the center from the very beginning. Increasingly, kids either walk into clinic or walk out of the clinic experience, not immediately, but with a genetic diagnosis. And these are things that we can think about as actual diseases where we're beginning to understand pathophysiology. And I'll talk toward the end of the talk about biomarkers that are not yet clinically useful, but might give us ways to have an anchor. So when we talk about treatment development in autism spectrum disorder, if we're talking about autism spectrum disorder as a whole, we're talking about a kid that maybe fits this pattern, which would be something like Fragile X syndrome, a kid who fits this pattern. I'll talk about this sort of pattern at the end. And if it's a well-powered study, about 148 other kids who all fit different patterns, and then we're saying, well, we've got a treatment that addresses autism. And you'll note that even in the center, you don't see perfect overlap. But when you start thinking around the edges and when you start thinking about disease models, certainly we're not talking about treatments for a disease. We fairly recently actually have genetic data, just in the last few weeks, that fits this model to a larger degree, which is what the epidemiology fits. The idea that there are likely multiple risk factors, both genetic and environmental, that coalesce in an individual to lead to substantial risk of autism spectrum disorder. But each of them individually probably contributes a very small amount of risk, and that at least is supported on the genetic side with common variants. But some of these by themselves probably do very little. Where we've seen much more traction from people like Dan, Matt State, and others are rare events, either genetic or environmental, that contribute the majority of the risk for an individual. These are things like Fragile X syndrome, which I'll talk about some in a bit, but also things that have been identified within autism spectrum disorder, so within groups of kids who are defined as having autism, and then there's a genetic risk variant that's identified, and then we begin to understand the syndrome in the larger group of kids, as well as things that weren't originally thought of in relation to autism spectrum disorder, like tuberous sclerosis or P10 hamartoma syndrome, but we now understand robustly increased risk of autism. These continue to emerge, including lots of single genes that can be implicated. As these emerge, you also see some examples on the environmental side that are not maybe as tractable, but are similarly robust, like exposure to valproate in utero. We can hope that some of these will coalesce around common systems. The example, and I'm not going to spend more time talking about today, but tuberous sclerosis and P10 hamartoma syndrome coalesce around mTOR signaling. There are some other ideas of genes that may similarly impact that signaling pathway. And you would hope then that in some cases that would turn back to common risk variants in the broader population, where you might have something that you could study in one of these rare variant populations and turn back into the broader group. Thus far that hasn't been borne out, but we can hope for it. That's actually the simplest level at which we can understand risk, however. So there, right now, you could say there's more than 100 genes that you can implicate. If you include copy number variants, it increases further. But as you go up this cascade and across development, things get dramatically more complex. So that by the time you get to synapses, we're talking quadrillions. And behavior essentially is infinite, right? But we've defined the disorder at the level of behavior. And so you should see things coalesce near the top. If that's true, you can imagine that you could have a treatment that's based on convergence across pathophysiologically anchored studies, but at the level that underlies behavior. And that's really the goal, the ideal. Whether or not that's realistic across all of autism, I would say maybe not. And most of the coalescence that's been described to date is at the level of protein networks and to a lesser extent cells at a particular time point in development. It's hard to step up much past that, although there are isolated examples. And that leaves a big gap. Because we've defined things at the level of behavior, we can imagine that you can jump from an understanding of proteins and maybe cells all the way up to behavior, but you don't necessarily know that because these pathways can go in lots of different directions. That's a dramatic improvement on every medicine that we use in the clinic to date, at least in the psychiatry side. It was developed based on the black box model without any idea of pathophysiology, and usually without an idea, initially at least, of what the medicine really did in the brain. It was based on observation. An additional challenge, this isn't just for autism spectrum disorder, but when you think about neurodevelopmental risk more broadly, is that now if you identify a gene disruption or a copy number variant that leads to autism risk by loss of gene function, oh yeah, I have to remember I can only see it up there, not on my screen, but this individual would have a deletion either of a large genomic region or a disruption in the gene. You can then go about looking for something that is a duplication of the same gene region, and by and large you find them, and by and large you find that they also increase developmental risk, which is really troubling. So if we had something that made this individual better, we might actually make this individual worse. You could still see convergence at higher levels, but that's troubling when we think about things that target what we believe is actually leading to risk. So that's what I'm going to say about risk of autism spectrum disorder. I'm now going to talk very briefly about the treatments we are confident work and how we got to that knowledge. Even though behavioral interventions are where most kids with autism should be spending almost all of their time, as opposed to on the medical side, I have one slide on it, and I'm not actually going to spend time there. But this is to say that we do have robust data suggesting that early and intensive behavioral interventions done in different ways help kids. Which kids benefit the most? We don't know that clearly. What fits what kid? We don't know that clearly, but clearly this is something that has impact. And then there are other things that can be added on in various ways to those interventions that also appear to have impact. And then there are more targeted behavioral interventions that have also been studied, although less comprehensive sorts of outcomes that people are looking for. All right. That's all I'm going to say on the behavior side. Forgive me, we'll come back kind of at the end. On the medication side, it's instructive to recognize what's happened in the clinic as opposed to what's been studied. So do folks know what medicine has the most placebo-controlled trials in autism spectrum disorder? Okay, so that seems like it should be the right answer. Okay, any other guesses? Methylphenidate also seems like a reasonable answer. So it's Secretin.
Oh, yeah.
7 randomized controlled studies. Does nothing whatsoever of any use, which is really distressing, right? So, and why was this done? There were a handful, less than a handful of kids who were reported to have dramatic improvements in language and behavior after receiving Secretin during a GI procedure. And that got a whole lot of attention. Likely tens of thousands of kids in this country received Secretin infusions, and then 7 randomized controlled trials were conducted. And it really doesn't do much of anything, but what was learned in the process is the placebo effect in autism can be quite powerful. So you can see here, and this is from probably the best or one of the best of these studies from Jim Bodfish and colleagues. The placebo has this huge effect, and they looked across multiple measures. This is a lumped-in measure of autism symptoms, but you can see that the placebo does better than Secretin, but the placebo effect was just enormous and present, depending on how you define it, in about 50% of kids. And then also instructive is even after told the results of this study, and this is one of the most stark quotes you'll ever read in a paper, the majority of parents were still interested in the treatment. And that isn't that these parents are dumb or foolish. It's because we don't have other things to offer. And when somebody says this could really make a difference for your child and is based on biology in some powerful way, in particular when it's targeting core symptoms, people want something. And of course they want something. But it's really hard to study medicines that outperform this sort of a placebo effect. Okay, so all of the medicines that have evidence in support of their use are treating associated symptoms. And where we have FDA approval for irritability and agitation, we have 2 medicines, Risperidone and aripiprazole, that from what we can tell don't do anything that's specific to autism spectrum disorder. So if I was up here and agitated, it would be just as reasonable to give me either an oral or injection dose of one of these medicines to help me calm down. They are medicines that are fairly universally calming and should be reserved for cases when kids are really agitated, not for cases when kids are just grumpy. But you're looking at medicines that also have pretty substantial side effects, not things that we really like to use. And then we have reasonable evidence for essentially each of the classes of medicines usually used to treat ADHD as also helping kids with autism who have ADHD. And that's it. That's the sum total of data that's replicated supporting treatments in autism spectrum disorder on the medical side. A little grim. So I'm going to talk about first the precision medicine path, and I'm going to use the example that has been around the longest and has carried forward the furthest. That doesn't mean it's the first that will lead to a treatment, but it's the example of Fragile X syndrome, and this is just to note that there are other examples that are currently under study and could be worthy of discussion here. But I'm going to use the example of Fragile X, in part because there is now quite a lot of data both identifying the rate of autism in Fragile X, but also helping us understand pathophysiology of this as a disease that can be studied. So autism, I would say if you didn't know these kids had autism spectrum disorder, maybe 25–30% of them would be lumped into the general population of folks with autism. If you didn't know they had Fragile X syndrome, probably you wouldn't distinguish them as particularly different. Once you know they have Fragile X syndrome, they are different, and you can pull them apart as having less repetitive behavior and some other different features. They all have mild to moderate intellectual disability. Many of them have hyperactivity and impulsivity as a major problem during early childhood. Some of them have seizures. They have a characteristic facial appearance. They look a lot like me. Long face, prominent jaw, prominent forehead, getting more prominent over time. Well, that's not characteristic of the syndrome, but this is to say we don't necessarily pull them apart based on appearance, at least not until after puberty. So the genetics has been known now for approaching 30 years. Expanding trinucleotide repeat in the 5' untranslated region leads to loss of the protein product, FMRP, which is an RNA chaperone. It has multiple different roles in neurons, and this is the one that sort of has the most data behind it, but certainly not the only potential pathway toward understanding pathophysiology in a way that would lead to treatment. But if this is your presynaptic neuron releasing glutamate, we usually think about signaling at AMPA receptors or NMDA receptors, but it also signals at metabotropic receptors, including mGluR5, that in some ways serve to tune the synapse up or down. You see increased FMRP translation downstream of mGluR5 signaling, and you also see with mGluR5 signaling a removal of AMPA receptors from the recirculating pool. If you have intact FMRP, you actually see it acting as a brake on the action of the mGluR5 receptor. If you don't have that, you get excessive downstream signaling and removal of the AMPA receptor from the recirculating pool, as well as a cascade of other events, with the idea being that you could potentially modulate the system. Amazingly, you can actually see this under the microscope with excessive dendritic spines that are not suppressed by a mature synapse here. You also can measure this electrophysiologically with excessive long-term depression. But amazingly, if you have postmortem tissue from somebody with Fragile X syndrome and tissue from a mouse that is lacking the FMR1 gene, you see the identical phenotype at the level of dendritic spines, where you see here excessive dendritic spines that have this long filopodia-like appearance, and here you see a typical dendrite with normal appearing dendritic spines. That's great, that's wonderful. The mice really don't behave in a very abnormal way. So they're a little bit hyperactive, not dramatically so. They have some social deficits, but they're in tests that don't easily translate across species. They have impairments in learning that are very subtle and have been difficult to replicate across labs, at least those that would more easily translate. They do have inducible seizures, so that translates a little more easily. And skipping over a couple decades of work, following this hypothesis, a group from Roche, and that's one of the, although I was not involved in this study in any way, it's one of the disclosures, was able to test the equivalent of a human clinical trial, was able to deliver a drug that tunes down response at the mGluR5 receptor shortly after weaning for 4 weeks, which is all the way to sexual maturity in a mouse. Obviously, that's not terribly easy to translate back to a human study, at least not directly, but importantly, they did see improvement in some behaviors. And they saw a rescue, really quite dramatic rescue of the dendritic spine phenotype as well as electrophysiology. There were no obvious ill effects on health. And this is about as far as we've gotten up this cascade, where there is still a gap. And I would argue, actually, because we can't easily translate the behaviors that are studied in this particular mouse model into humans, the gap is really a black box from looking at electrophysiology, anatomy to some extent, all the way up to what behaviors we would expect to change. And that's where the story has been interpreted as really depressing, and it is in some ways. How many people in this room would say, with these data, the first thing we should do is test whether mGluR5 negative allosteric modulators work in adults with Fragile X syndrome? Okay. How about adolescents? Okay. So that's what was done first, and that isn't because people were stupid. It's because you actually, for safety reasons and because of regulatory requirements, you have to start in adults and walk down to adolescents and walk down to older school-age children and younger school-age children until you finally get into little kids. Most of us would want to test something like this that seems to affect synaptic plasticity in an age range where you can actually probe learning in a predictable way. But you can see in these adult studies, and it's hard to see, but in gray is the placebo. And more times than not, the placebo outperformed the active medicine at 3 different drugs in this study, which is discouraging. And it was sliced and diced in different ways. There really isn't much of a signal. Some hints of a signal in things like eye tracking, but not terribly robust. And both Roche and Novartis, who had research programs around Fragile X syndrome, dropped their research programs. That's a pragmatic decision, but sort of a discouraging one. So it could be that this mGluR5 mechanism isn't the right thing to study. And I think that that has to be one of your hypotheses, particularly if you're a drug company investing $1 billion in this sort of thing. But there are other things that could explain this, and I would say certainly the challenge of the placebo effect is there. But I would say that the big things that you would guess would be you're studying the wrong age. You're studying an age at which you're going to have a hard time finding the outcomes that you're interested in. And then the outcome measures used, which were aberrant behaviors lumped together, are probably not the ideal outcome measures for something that affects synaptic plasticity. Dan?
Maybe we should discuss this in the discussion period. I'll leave it up to you to punt this or not. But I mean, kind of fun, and fundamentally, because you've studied the mice and the patients been involved, my sense as an outsider is that the mice have very little of the deficits and and their memory improves, right? When you give them the drug, memory wasn't tested in these kids.
Right. There were some attempts, but it was not well probed.
Aberrant behavior was, and there isn't much behaviorally going on in these mice, so that just the parallels are totally missing. That would be my sense as an outsider.
So essentially,
there's a lot of wishful thinking that went into the study design. A lot of that, I would say, probably driven by certain investigators who felt that actually behavioral parallels aren't that important. And maybe this is teaching us that maybe they are.
Well, so I think there are a number of pieces there that, and we could come back to a more detailed look at that, but I think part of this was just simple pragmatic decision making on the part of the company or companies. And there were quite a number of people consulting who said, really we're not going to know until we get down into little kids, but you have to do safety work in adults first because these were not drugs that had previously been tested in large populations. Part of the pragmatic side is about picking outcome measures that are known to be robust and that the Food and Drug Administration or the European regulatory agency has approved in the past. So they picked the aberrant behavior checklist because that was what was used for risperidone and aripiprazole. Pragmatic decision, you hope you see an effect. And then they had some preliminary data that suggested maybe they were going to see a change there. And so that they extended into larger samples. And in some ways, pragmatic decision-making isn't unreasonable. But if you just back up and say, what would we ideally like to do? It never would have been done this way, right? And I think that that's important. There are also differences between expression of the Fragile X protein in primates and mice that are likely quite relevant. Work from Ninad Sestan showing that you have changes in cortical expression of FMRP across development in primates that are not seen in the mouse. So there are some important places to be hesitant there. But now, importantly, a group of investigators funded by NINDS and led by Liz Berry-Kravitz as well as a group more on the outcome measure side at the Mind Institute, has a study funded to look across development, so looking at 3 to 6-year-olds, which makes us a little nervous, right? But the pharmacokinetics and initial tolerability was done already down in that age group, and actually looking at language learning as the primary outcome measure with a parent-mediated speech and language intervention, so that there is a baseline upon which you would hope that the medicine builds, and looking at eye tracking as well as some EEG measures in response to auditory stimuli to understand whether we can drill down at least a little bit to see if there's an impact there. Whether or not that works, I don't know, but this is more like how you would actually want to test the hypothesis. There are lots of other targets we could talk about, and I'm not going to talk about today. So if this sort of approach works, I would say the thing that you would be most sure of, if it works, would be a benefit in a given syndrome. I would say that the other piece that we can hope for is that you learn how to study something. So I would say in that study, the hope is that this would be a paradigm that would allow you to get an actual yes-no answer to whether that hypothesis is a meaningful hypothesis to impact humans with Fragile X syndrome during a period of development where we can monitor something that ought to be sensitive to synaptic plasticity. And then there is also the possibility, but I would say at this point you have to be a little nervous about it, that would extend into a broader population with ASD without Fragile X syndrome. And I would say in this case, because it's Fragile X syndrome and we're talking about language learning, it's not necessarily just ASD, it may be intellectual disability without autism that could be a population where you'd see things extend. But I'd be thrilled to see that happen. I just am not counting on it. So that's one pathway. The other pathway in some ways is sort of the opposite. So when we see kids in clinic right now, there isn't really anything that we're doing based on knowledge of the pathophysiology of that particular kid that impacts our treatment. Often as a child psychiatrist, the first thing I do to make kids' lives better is I treat their constipation or find somebody who can, right? Very predictable, that will make their life better. If they have epilepsy, I make sure that there's a pediatric neurologist who is appropriately treating their epilepsy. And that is the first way that you're going to make an impact for a kid with epilepsy. We would never do a randomized controlled trial to see if successful treatment of epilepsy improves learning or autism, right? But we all believe that it's true. You then think about, obviously, language interventions. You think about interventions that have been shown to impact on IQ. And then as a child psychiatrist, usually the things that we're thinking about on the medicine side are not autism spectrum disorder, but are really symptomatic treatments that can have a big impact on the child's life. And then you have things within what we think of as core symptoms of autism spectrum disorder that are not actually going to help every kid and might be in the same domain, symptomatic treatments. So we have kids who come in to see us who just can't stop touching our faces in ways that are experienced as socially intrusive. Making that child more sociable is not going to improve their autism, but for another kid who is not at all interested in social stimuli, that could actually lead to a big improvement. So treatments in this sort of symptomatic domain, I would say, are important, and in some ways are things that might be a group of kids with autism spectrum disorder, but are going to make a much smaller impact. And you can come up with lots of examples of this. And we're starting to have places where you could say, well, you know, if I could impact the way the child takes in information from a behavioral perspective, or if I can go one level deeper and I have some understanding of a circuit that's relevant, maybe I can target a treatment there without really connecting to pathophysiology. And I'm going to use oxytocin as the example in this domain. So it has social impact across species. That's well described. Really no question about that. If you squirt some up my nose, I'm a little more likely to buy a car from you. So that's not a treatment for autism spectrum disorder. And most of the data with oxytocin is based on single dose administration, usually in a laboratory setting that is quite abstracted from real life. There also is some interest in vasopressin, which I'm not going to talk about, but these are sort of reciprocal hormones. Oxytocin is better studied. It seems to have more impact in women. Vasopressin may be a little more impact in men, but we could talk about that again after. there's really not great evidence that oxytocin is involved in some profound way in the pathophysiology of autism spectrum disorder. You do find places where there is some evidence. So Dan had some exciting data in the CANTNAP2 model. There's some data in the SHANK3 model. But there isn't clear evidence that across autism this system is involved in pathophysiology. But there is some laboratory data. This is from a French group and Daria colleagues using a laboratory task showing that oxytocin makes a person with autism more likely to play a reciprocal game of cyberball, playing catch with somebody that they think is an actual individual, versus a placebo squirted up the nose where you see no such advantage. And then Kevin Pelfrey's group, when Kevin was at Yale, showed that even though they did not see a behavioral difference, that the group receiving oxytocin versus the placebo group in response to either faces, or in this case eyes, or vehicles as sort of a visual control for that, showed activation in brain regions that could be interpreted as quite relevant for social function, including the nucleus accumbens, as well as quite a number of other regions. So you can change behavior, or you can change fMRI bold signal. And now and I'm showing up sort of the most encouraging of these studies. There have been quite a number of pilot studies, so not well-powered studies, usually done for 4 weeks, sometimes 8 weeks, looking at whether oxytocin squirted up the nose once or twice a day improves social function by various measures in autism spectrum disorder. This study actually showed a significant effect if you controlled for baseline oxytocin levels in the plasma. Interestingly, you saw that in the placebo group, you also saw a change in oxytocin levels over the course of treatment. And that's been described in a few studies outside of autism, that the placebo effect may actually be in part mediated by the oxytocin system. That's going to make it really challenging to study this, but intriguing. And we now would not advocate immediate incorporation of this, although there are probably tens of thousands of kids in this country who have been exposed to intranasal oxytocin. But we really need well-powered studies to understand whether this has an impact. We just wrapped one such, and at the Center for Autism and the Developing Brain, we were one of the sites. Because of that study from the Stanford group, which I should have called out earlier, Karen Parker and Antonio Harden and colleagues, we're awaiting plasma oxytocin levels to be able to actually look at that and incorporate that into the analysis to understand if there is an impact there. So I would say more data is pending, but we really need well-powered studies to draw conclusions instead of a series of pilot studies, which is what we have so far. Yes?
I have a question. I'm just curious, like, with these patients, you have 290 patients. What is the inclusion criteria? I mean, autism is on the spectrum disorder, right?
So it's very broad, and this is one of the challenges. So we tried to include as many kids with intellectual disability as without, and it went from 3 to 17. At our site, we did not enroll. We may have had a kid in that younger age range once everybody else had them in already, and we had months of safety data. But most of the kids in this study were older kids. It's one of the challenges that in some ways you imagine that the impact might be larger the younger you go. But for me, it doesn't feel ethical to enroll a kid until you've seen whether they're going to make dramatic gains with early and intensive behavioral interventions. So you want to be sure that they're already getting appropriate interventions and you have a chance to see how much they're benefiting before you make a decision about whether something like this is reasonable, even on the research side.
So what age are we talking about?
What age? It was 3-year-olds to 17-year-olds.
The ones that you have in this.
Yeah, but most of the kids were in the older age range. Only toward the very end of the study were the younger kids enrolled. So I would say one of the core challenges is filling in this sort of a gap between the 1% or less, and in most cases less, that may benefit from precision medicine, and this sort of broad impact on symptoms, but usually not very closely tied to pathophysiology. And I'm going to give an example of how these data might work. This is from an actual study, but I am not, again, advocating use of a related medicine because arbaclofen is not available. I'm not even going to talk about the putative mechanism here. but just highlight the data. So in this study, 150 kids, this is what's accepted as a well-powered study. The primary outcome measure was social withdrawal, measured on that same aberrant behavior checklist, parent checklist. No significant effect. I'll show you the actual data in a second. And then on one of the key secondary endpoints, clinical global impression of severity, there was an effect that's sort of right at that threshold of correction for multiple comparisons. And I think it's important to approach that with some skepticism. But to emphasize the skepticism, can you even tell that there are two lines there? Change from baseline in the primary outcome measure. There's no difference whatsoever. And then if you zoom in on the clinical global impression of severity, which is this simple Likert scale, the clinician rating of how severe the child's overall autism symptoms are, again, depicted as change from baseline, you see that there is some separation over time. If you look at the kids who are separating, there's a difference here seen in the no change condition, where you see an advantage for placebo. And the kids who are driving the effect here of this downward drift are kids who are really shifting quite a lot in terms of clinician ratings. That makes me nervous because we don't usually see those sorts of changes in the clinic. It's a small group, so it's about 15% of the overall kids in the active treated group who are having these big shifts. But if we had something, and I'm not saying this is it, but if we had something that improved something related to pathophysiology in a subgroup, and we applied that in the overall group of kids with autism, this is the sort of thing that we're going to be stuck with, right? Where you see a subgroup that's improving, and unless you know something about that subgroup, you really don't know how to carry things forward. So in this particular subgroup, it seemed like they had higher IQ and better language, which might relate to the outcome measures that are better able to pick up change in those kids. What we really would like, though, is some sort of a biomarker that says this kid may benefit from this treatment. With arbaclofen, people are looking at EEG now. There's a study funded, actually two separate funding streams. The EU aims to trials, as well as the PON mechanism, so Europe and Canada, looking at whether they can find that subgroup in a larger study that's well-powered. We don't know yet, but you would like a biomarker that obviously can be measured reliably, is replicable, but there are other things it could have that are not necessarily critical for carrying a biomarker forward for the purpose of treatment development or identifying a subgroup. So it doesn't necessarily have to be tied to genetics, although that would be nice. It doesn't have to have the lovely quality of being bimodal, although again, it would be nice to know exactly where the line is. It could change with treatment, it doesn't necessarily have to, and it doesn't have to be tied to pathophysiology in a way that we understand. But what we really want is that it fits in this gap, right? So it fits somewhere between precision medicine, big impact of treatment, theoretically, we don't yet know, and these sorts of symptomatic treatments that are really not satisfying. Most of us, when we see kids in clinic and are talking about medicines as options for their children, are disappointed. That's what drives us into the lab. We want something that fills that sweet spot. So ideally somewhere around here where you have a large enough subgroup that you can study it and that you can say you've really got something to offer, but you're making a bigger impact. There are different ideas of what might carry us forward, and I would say that these ideas are all fairly reasonable. On the genetic front, there are observations that a lot of the genes implicated in autism spectrum disorder, as well as genes with expression changes in various different studies, are bound by FMRP. So maybe a drug that impacts people with Fragile X syndrome would help a larger group. Unfortunately, we don't always know whether FMRP is leading them to increase or decrease in terms of their translation. So I would expect that this is going to be one of the challenges around our inverted curve. There are some neuroimaging biomarkers that have been proposed. I would say the EEG is probably more feasible for the larger group of kids with autism spectrum disorder. We don't know yet how that's going to translate, but that seems like it would be a very useful sort of outcome measure, as well as something, meaning intermediate outcome measure, as well as something to identify subgroups. And then there are other ideas, and I'm going to talk about one, again, choosing something that corresponds to something I've worked on, so that's no secret, but also choosing something that's been around for a long time so we can talk about something that has quite a lot of data behind it. So elevated blood serotonin levels in autism have been described for more than 50 years. And this is a Dutch study, so you can tell from my name, maybe that's a bias too, where in white is what would be a bell-shaped curve in the general population, and in this sort of yellowish color on this particular screen, you see in this study a second peak of blood serotonin levels. Now, the serotonin levels in the blood, certainly not in every kid, it's about 25 to 30% of kids. The serotonin, that's interesting. I don't know that black box. Well, in any case, it doesn't show up on my screen. But the serotonin that's in the blood is packaged in platelets. It does not cross the blood-brain barrier. This is not something you would imagine as having a direct effect. It really is a peripheral biomarker. In the platelet, it's taken up via the serotonin transporter, which I'm going to talk about in a little bit. It also has mechanisms that impact its release. It's not synthesized in the platelet, it's synthesized in the gut. So there are sort of a complex cascade of events probably contributing to blood serotonin levels. But importantly, this is much, much simpler. It's a reduced system in comparison to the serotonin system in the brain. And also, blood serotonin levels are more heritable than autism. And so you can actually use that to map genes potentially. There are some linkage peaks overlying the serotonin transporter. We don't do linkage anymore, by and large. We have more powerful techniques that have yielded actual genes. But in these studies, you found a signal in males but not in females. If the family had one or more affected females, you saw no signal whatsoever. I'm not going to spend a lot of time talking about this genetics. I'm actually going to end up taking a fairly abrupt right turn in just a moment. But the linkage signal in one population at least had a number of rare variants of the serotonin transporter underlying it. In each of those rare variants, you saw an increase in serotonin transport that led us to look at the particular signal in association, and there was some hint of association with rigid compulsive symptoms and sensory aversion, which led us to ask a question I'll tell you about in a moment. Importantly, in these families, you typically saw transmission from an unaffected mom to a couple of boys, and often a couple of girls who did not have autism spectrum disorder. You also see that some of these variants are found in boys who are unaffected, and these rare variants are not implicated in the de novo variant story and don't have a robust signal in the overall case control association studies in autism. But now, almost 10 years ago, actually 10 years ago, we took the most common of these variants, the ALA56 variant. We made a mouse model to understand how it affected the serotonin transporter in a mammal, but also to understand how that might contribute to autism risk. At that point in time, these mice grow normally. They recapitulate the biomarker, and then I'm going to skip over a whole bunch of data to tell you that because we were interested in that sensory aversion phenotype, and because the serotonin transporter is transiently expressed in sensory regions of the brain during development, we were quite interested in understanding whether the activity of the serotonin transporter in the thalamus at a key point in development might relate in particular to the tactile hypersensitivity that you often see in kids with autism and seem to drive that phenotype. So we had the hypothesis that a change in serotonin signaling during development was going to impact formation of sensory-related brain structures and then lead to that sensory aversion phenotype. We initially looked later in development, and this is hard to see on this screen, at the whisker barrel cortex, which can be seen beautifully under a microscope and allows you to do comparisons from animal to animal, where we saw really no difference in structure of that cortical region in animals actually a little later at P21, postnatal day 21. We then wanted to look earlier during the period of time when this transient expression of the serotonin transporter is really clearly relevant for projections from the thalamus into the cortex. And so when we did that, we initially did it the right way, which is that we looked in animals, embryos that were from heterozygous dams bred to heterozygous sires, so that you're able to control for the parent genotype when you're looking at the genotype of the offspring. And then we're looking at embryos that either have the mutant genotype or the wild type genotype to make a straight comparison. That's the right way to do the experiment. My graduate student wasn't getting enough animals quickly enough, and he wanted to know whether this was going to pan out or not, and I was behind the idea, so he also did some of these breedings the wrong way, which is to just breed some of the wild-type animals from the colony with some of the mutant animals from the or some of the mutant animals from the colony, so that you have all wild-type offspring or all mutant offspring that you can harvest as embryos and look at potential differences. And he, in the initial analysis, put these different sorts of animals from different breeding schemes together and saw what looked like a broader breadth of trajectories from the thalamus projecting into the cortex in the mutant animals compared to the wild-type animals, and got really excited. But then as we looked at the data, it was really inconsistent, didn't look terribly robust. So then he split the groups in order to add animals and understand whether this was something that was robust when the experiment was done the right way. And when the experiment was done the right way, it was really disappointing. There was no effect whatsoever. But instead of just dropping it there, he also did more of the animals the wrong way, and just looking at an embryo from each, so that you're able to control to some extent for the background of being in a single pregnancy. And what he saw was that the effect was present when the maternal genotype differed, and the effect was not present at all when just the embryo genotype differed. And this was primarily in the rostral projections, where the serotonin system is an important modulator, and not in the caudal projections, so something that serves to some extent as an internal control. This coincides with data from Alex Bana and Pat Levitt, showing that in a critical period of development, the cortex is receiving most of its serotonin actually from peripheral sources, from the placenta, with the potential that the maternal system could impact the placenta. So when those projections are starting, you're in a period of time when actually the axons from the raphe have not yet reached into the cortex. And so the source of serotonin from the placenta is still quite relevant to the developing forebrain. And then as you march forward, that's less and less the case, to the extent that by the time the animal is born, there's very little impact of the placenta just before birth. So we looked to understand whether the maternal effect could be seen in the forebrain, and you would hypothesize then not seen in the hindbrain, where there is an embryonic source of serotonin, and saw exactly that. You see a maternal effect in the developing forebrain. You see an effect that is nicely paralleled in the placenta, and you see no such effect in the hindbrain. And then when you look just at the embryo genotype, you see no such difference consistent with that. And then I'm going to, again, jump over a whole bunch of work that we're still doing, but have done, to try to understand the mechanisms underlying that in the mouse. But because at the end of the day, at the beginning of the day too, I'm a child psychiatrist, I wanted to understand whether this was a neat thing that happens in mice, or whether this is actually something that's relevant to humans. And that's a little bit of a challenge in a number of ways. One is that if you're interested in this particular rare variant, it's rare. And so you're going to have a hard time getting a population to study. There are quite a number of other variants in the serotonin transporter gene, but enough that it's a little hard to do a computational analysis that puts their different effects together. However, we have this biomarker, right? So we've been looking at blood serotonin levels in people with autism for more than 50 years. Lately, we don't look at it a whole lot. But we thought if we could get our hands on a population in which there were levels in moms, levels in dads, and levels in offspring, we could look to see if there was any effect of mom's biomarker in association with child's phenotype. We didn't have access to a sample that had serotonin levels during mid-gestation, which would be where you would imagine the effect, if there is an effect, would be relevant. We also didn't have maternal serotonin levels in a general population sample, but what we had access to from Ed Cook at the University of Illinois at Chicago was a sample in which he had measured blood serotonin levels. Actually not he, but others he had collected the samples and directed the study, measured serotonin levels in offspring, as well as mom and dad. So if you think about that study design, it's important to note that this is heritable, and if you just look at straight correlations, you do see significant correlations between levels, between offspring and parents. To a lesser extent, parents' serotonin levels actually correlate for reasons we don't completely understand. So you must correct for any significant proband effects. And then what I think is that the dad's levels can actually serve as something of an internal control. You wouldn't imagine dad's serotonin level has any effect on the developing embryo. So we, in an initial look-see, and this is sometimes what happens when you don't actually imagine you'll have a real effect. I told Ed about the findings and asked, could you look at your population? And we didn't talk too much about how we were going to do it. And they ran a correlation matrix. And they looked at dads and they looked at moms. And we already knew that the offspring effects really weren't there. And what they found was consistent with previous data showing that there was no robust correlation between the offspring serotonin level and any phenotype in the kid. Remember, all the kids have autism. But then when you start to look at individual measures, and this is just social scores, not CSS, not clinical severity, CSS, clinical severity score. So you saw an effect in mom, you didn't see an effect in dad, you saw an effect in repetitive behavior, and then in different subgroups, because some had the data, some didn't, PPVT, as well as expressive, so receptive and expressive communication measures, Vineland adaptive behavior skills. So at this point, it's getting a little spooky. And I should say that there were some measures where it was not significant, but everything was more or less in the same direction. But this is not really the right way to do this. You don't just look at every measure. This happened to be the thing that was done sort of inadvertently in the initial look-see. So we wanted to do a data reduction method to try to have a legitimate statistical test. And so we did a latent class analysis, and we included the robust measures that didn't overlap too much with other measures across this particular population. And we found 3 classes. And the things that we included in that latent class analysis included nonverbal IQ as well as adaptive behavior, measures from the ADI and measures from the ADOS. Now calibrated severity score as opposed to just looking at scores within domains. And you can see that these groups separate. By and large, it just looks like severity, although you can see in some places the separation appears to be different depending on the particular phenotype that's being considered. And then we did the simple test over again, but in much reduced data where we were looking at blood serotonin levels now between those different latent classes and found a significant effect that was driven almost entirely by this most severe group that had lower blood serotonin levels in moms. You saw no effect of, no association with dad's blood serotonin levels and no association with proband serotonin levels. What that means, I can't really tell you. So there's a lot left to do on the mouse side around mechanism. On the human side, the first thing is to replicate, but also to extend. So we need to know if this relates in any particular way to autism or whether this is just a neurodevelopmental effect that you would see in any population. We also would like to see this prospectively. So the blood serotonin levels were measured in moms after the child was diagnosed, certainly not during the time period when they'd be relevant. And then from a mechanism standpoint, we do know from work, again, from Alex Bonin and others, that the levels of serotonin from the placenta really do impact the projections coming from the serotonin neurons in the developing embryo brain. And then there's now some really exciting data from Rob Malenka's lab showing that the serotonin system interacts with the oxytocin system to impact social function in mice, and that you can perturb the serotonin system, serotonin projections to the nucleus accumbens, and disrupt social function. You can also drive social function by. And then there's lots of questions about how you'd actually carry this story forward. In some ways, you've humored me through my own restricted interest. Importantly, I would say that when we think about something like this, and I would say the same thing might be true for the maternal antibody story that's emerged from Judy Vanderwater's work that I would say is not similarly ready to translate. But it may be that the biomarkers that we're interested in might be things that we can't measure in the child at the time point where we're looking, and it may be that this sort of a biomarker will then tie to another biomarker that we can measure in kids in real time. I told you I'd come back to this at the end, so our ultimate goal is to tie things together from a biological intervention perspective and a behavioral intervention perspective. This is the only example in autism to date where there's such a statistically significant effect, at least published. And this was a study where both groups here, shown in blue and in pink, were receiving risperidone. And the group in blue was also receiving what now most of us call the Ruby parent-mediated behavioral intervention. There is a statistically significant effect, but if you go 6 months out, there no longer is a significant effect. Importantly, the group in blue was taking a lower dose of risperidone, which was allowed to float based on the clinical judgment of the psychiatrists, which is a challenge. So it is really hard to do these sorts of studies where you put two different interventions together. The ideal is probably something more like what's being done with D-cycloserine and extinction learning. Clearly has an impact in mice and rats. And there is emerging data in human studies that it potentiates exposure and response prevention based on the same sort of extinction learning we can study in rodents, where you see that early in treatment you can see a more rapid response in the group receiving D-cycloserine. If you look at the end of treatment, if you've done as much treatment as you'd like to do, you actually don't see such a big impact anymore. You can imagine if we had something like this, not that it's D-cycloserine, in autism spectrum disorder that would really leverage our behavioral treatment to have a bigger impact, that could really make a huge difference. So that sort of thing is, I think, what we'd eventually like. So if you put oxytocin together with, not that oxytocin works, together with social skills training, could you really leverage that social skills training to have a sustained impact over time? So because I talked a little longer than I intended, I'm not going to read my summary slide, but suffice it to say that autism is not one thing, and we shouldn't expect one treatment that's going to affect everyone, at least not if that treatment is anchored in pathophysiology. Placebo effects are a real challenge, and they're a challenge for us on the behavioral side, just like they're a challenge for us on the medication side, but that we do have pathways forward, and that when one of these pathways opens, we're not just going to have a new treatment, we're also going to have a new understanding how to study a treatment that I think will be impactful beyond just a single potential medicine. I want to thank folks who are involved in our baclofen study, as well as folks involved mostly on the Vanderbilt side before we moved, as well as folks involved in some of the work I showed you very briefly from the mouse and then translated into the human, as well as the funding mechanisms we've cobbled together over the years, mostly not to fund that maternal serotonin story, although we are fortunate to have a new grant that will help us do just that. So thank you very much, and happy to take questions. Yeah.
So it wasn't clear to me. What the phenotypes were in the mouse model for the serotonin alpha-56.
Yeah.
So what were the phenotypes that you see in the mice?
So the mice, if you look at the basis of the offspring phenotypes, not with regard to the maternal genotype, but with regard to the offspring genotypes, we see that those mice have a repetitive behavior, so they go up and down from the cage lid. It's not terribly stereotyped. I can't say that I understand what they're doing or why, but they do that. They also back out of the tube test much more frequently than the wild type mice when they're confronting each other in that context. They also have a change in ultrasonic vocalizations at P7. That's all with respect to the offspring genotype. So we're just now starting to do some of the behavior with respect to the maternal genotype to look at those effects. Those studies are challenging in part because you can use one offspring per litter, and so having the money to do some of that is hard. But yeah, so we don't.
Social.
So the social in the tube test, yes, and
Just on the tube test, but not
on the tube
Crawley
test. In the Crawley test, so if you control for the amount of locomotion they do, so if you exclude the animals that don't fully explore the apparatus, they have a difference. But I don't find it a very tractable behavioral test in this genetic inbred strain background, because they're on a 129S6, 129S4 strain. We're getting obscure, forgive me. And so about a third of the animals, regardless of genotype, don't fully explore the apparatus. They just go to a corner and sit there. No matter what I say to them, they still go to the corner and sit there. And it has nothing nothing to do with genotype. I can tell you so if we had just stopped studying the behavior when we had a significant effect, we wouldn't have realized that. Because the first group of 8 animals per genotype that we ran, we saw a significant effect on the Crawley test. But when I looked in, I didn't feel like the animals were really exploring the apparatus well. We ran more animals, and then you only see the effect if you exclude those that freeze. We could talk more about that test, but probably isn't in everybody's interest. We can talk about it this afternoon. Dan?
Yeah, I mean, just that raised a question, but so I guess I have 2 questions.
Yeah.
But wouldn't it make sense then to, I understand the use of genetic background as a control, it makes it easier, but given all of that, wouldn't it make sense to kind of outcross and look at F1s? Because my sense is that you're going to find things that are more likely to be translatable to humans when you can see it on multiple different backgrounds. And we've seen that in our studies on neurodegenerative disease. So that would be just one kind of statement. I'll let you, you probably, hopefully you agree with that, but if you don't, I'd be happy to talk about it more. But the question I had,
Try to remember that on one finger. Go ahead.
Okay. So, but the real major question is around, the serotonin study, which is really quite fascinating and, quite relevant. And it makes, I remember, this is just a funny anecdote, but the first time I ever met Portia Iverson, I remember I talked with her for about 3 hours, and I didn't know much about autism at all. And I just looked at her and I said, God, just sounds like serotonin to me, at that point, because of, when the gut problems and everything. But the epidemiology, there's some kind of connection with depression. I don't think the data is very clear. It's certainly not convincing to me that mothers who take that the SSRIs themselves have any effect. It's more kind of related to the underlying, and that could be polygenic risk. But I was just curious if you'd think about that, because SSRIs would increase serotonin, and so should increase blood serotonin, and so therefore would kind of have an opposite effect, might actually be protective in some way, like you give maternal SSRIs. I'm just, thinking kind of slightly tangentially, but I'm just wondering what you think of all of that and the epi kind of the human connection to this.
So I'll take the mouse question first. Okay. So with regard to the mouse inbred strain, we made these on one background. We actually did back cross them to C57. We did not do the F1 cross. And actually right now we're not really focusing on the offspring genotype. So there's a reason I didn't sort of go over the behavior based on offspring genotype. We're really focusing on the maternal effects. Interestingly, and although maybe just interesting to me and maybe you, Dan, when we back crossed to C57, we lost the effect on the serotonin transporter's function. There is a genetic variant in the serotonin transporter in C57 black 6 mice. We actually back crossed the wild type 129 allele also into the C57 mice, but we don't find that they preserve the regulatory cascade that we think that this ALA56 variant affects, which was a real pain because that took us 3 years to do the back cross and all the behavior, et cetera.
That would be important to publish. I think that's.
We did publish it, yeah.
Okay,
but with a fair bit of despair, but we did publish it. And then with regard to the human data, so I don't read the SSRI data like you. I don't read the human SSRI data as consistent with a risk of autism related to SSRI exposure. There is now a novel finding, although we'll see if it holds up, that suggests that there may be more of a spectrum of risk with SSRI exposure and language development. But until it replicates, I'm a little hesitant, because if you just dial back 4 years, you would say the same thing with regard to autism. There is clearly a signal there if you don't control for the maternal history. So it looks like if you need to stay on an SSRI across pregnancy, you have a higher risk of having a child with autism spectrum disorder. So why do you need to stay on an SSRI throughout pregnancy? Probably you either have more severe depression or more severe anxiety or more severe OCD. And we really don't know from those studies which it is, but there also is abundant data showing that if you look at first-degree relatives of people with autism, you see an increased loading of quite a number of things, psychiatric, autoimmune. So there is something there. It just isn't that clearly related to medicine. I don't know, and I would say the simplest hypothesis is this is not going to be a maternal effect of this, an effect of the maternal serotonin system that is going to be autism specific, because I don't think that evolution or biology knows the difference between autism and not autism. I think it impacts on brain development, and that may relate to autism. And it may be that there's a subgroup of people who have other potential risk factors where it amplifies. The last thing I'll say about it is, I also don't know, because of the breeding scheme, and we're doing blastocyst, or we're anticipating doing blastocyst transfers in the near term, whether the embryo genotype amplifies the maternal effect in the mice. And that would be a really interesting thing to look at. We also don't have a big enough sample to be robustly powered for an interaction effect between the biomarker in the mom and the biomarker in the kid. It would be fascinating to know. I can't tell you at this point. And then with regard to blood serotonin levels and SSRIs, blood serotonin levels go to 0 or near 0 with an SSRI. So why is that? The SSRI blocks the serotonin transporter, so serotonin can't get into the platelet. And then the challenge is that we wouldn't imagine that platelet serotonin relates at all to the placenta. We think it's the serotonin that is released from the platelet and is sort of circulating in the plasma. But that's almost impossible to measure because the serotonin levels in the platelets are so high in comparison. And because when you try to isolate the platelets from the plasma, you end up inevitably getting some serotonin release. It's very hard to, with confidence, say what the circulating levels of serotonin are in the plasma. There are ways to do it, but most of the time when somebody publishes one of those studies, they get jumped on as likely not accurately measuring them, for what it's worth.
So are there any
Yeah.
changes on the serotonin levels during pregnancy?
That is a great, so I'll repeat the question for those watching from the video. If there are any changes in the serotonin levels in moms or moms-to-be across pregnancy. So the data that we have on that in humans is from like the 1960s and is not super consistent. It looks like from those studies that it's fairly consistent across pregnancy. From the mouse, we actually saw a change across pregnancy. And interestingly, although maybe just to me, the serotonin system actually is involved in the regulation of insulin secretion during pregnancy. So the increase in insulin needed during pregnancy is needed in part by the serotonin system. And there are transient expressions of serotonin receptors in the serotonin transporter in pancreatic beta islet cells during pregnancy that then resolves at the time of delivery, which is sort of fascinating. And we did one thing in the mice to see if we could play with that in relation to serotonin levels in the placenta. Didn't see an impact, but we're not sure that we did it in a way that we'd necessarily see that. Yeah. Yeah, Shafala.
That was a fantastic talk. And
Oh,
say what
thank
I
you.
appreciate about your work, and especially the Backlund paper, was that I think many times when clinical trials fail, there is this sort of sense of like desperation in the field, like, oh my gosh, another medication doesn't work. And I think there's a lot we can learn from those failures. And I think in that paper in particular, you raised a lot of the issues that you talked about today, like, we looked and there might be subgroups that respond better, or you're dealing with a heterogeneous sample, those sorts of things. So I think you've been really a thought leader in this area. I'm really interested in the placebo effect, and I just want to get back at that for a minute with you, because I think your finding with the oxytocin was really interesting to me, because I think there's still this sort of fundamental debate, and I'm sure it's not one or the other, as to whether is the placebo effect, driven by like biased, parent and clinician reporting, around belief in the potential effectiveness of a treatment, or is there a true change that's occurring in the context of being in a trial? We talk about the Hawthorne effect and, this sort of this true, benefit that can happen. Even in our infant studies, we think that some of the reason we see maybe attenuated symptoms in like our high-risk infants when we're, looking at diagnoses is that there's a surveillance effect because their babies are followed developmentally, and maybe there's an enhancement even of the maternal-child interaction and that environment early on that actually improves development. So I'm just curious, I know we think of it as a problem, but if it's really the fact that following kids in this way is therapeutic, it actually might be not a problem, but an opportunity that we need to understand better. Because if we can like exact, change and improvement without a drug, then, and the bummer is that it's like leading to all these trials failing because you don't see a difference between groups. But anyway, so that was sort of a long-winded way of just asking you what your thoughts are on that and what, just in terms of whether the placebo effect is truly a change versus.
And so this is, I have, obviously I have some embedded slides on the placebo effect. I did not talk to Shefali in advance. Could you please ask about the placebo effect? But we talk about placebo because it's one word that we can use that summarizes a lot of effects. And because we all think we understand what placebo means. But really, the placebo is a control for nonspecific change during treatment. It is not really an effect of the placebo itself, right? And if you think about what the placebo is, it's sort of the opposite of a waitlist condition. So if you're thinking about true placebo effect, when you have a waitlist condition, you're telling people nothing's going to change until you get off the waitlist. You have to wait for your active treatment. When you're giving the placebo, you're saying watch for change, right? You don't tell people that they're taking placebo. And there are a number of things here that I think, so part of it is nobody ever enrolls in a treatment when they're at their best, right? Never happens. So if a child seems to be doing well in the last month or so, and the parent has even been thinking about a clinical trial, the parent will decide not to join. And so we have this happen. We'll have a family contact us, and then they decide that they're not going to participate after all. And often it's because their child's doing a little better. And then if 3 months later their child's doing a little worse, they might reach out again. This is one of the reasons that people think that doctors do good, right? Because you never go to see a doctor when you're feeling well. And that's why bleeding seemed like a good thing. Either you're gonna, right? Either you're gonna live or you're gonna die. And if you saw somebody and you live, you figure it probably had something to do with that. So that's one thing. I think probably the bigger thing is that development happens regardless of whether or not a child has a developmental disorder. And if what you're doing is asking somebody, essentially, is your child developing? The answer is almost always going to be yes, or at least 50% of the time going to be yes. And that's really challenging, because if we are now trying to distinguish a little bit of improvement, we're now talking about a change in slope. And that's where actually this study in Fragile X Syndrome, this FX Learn study, that's explicitly what we're going after, right? These kids all have some exposure to enriched language with the idea that we're hoping for a change in slope. We're not imagining that those kids are going to be static. We're imagining that they're going to change over time, and we're looking for a difference in the amount of change. And I think that that's something that we don't do well. Most of our outcome measures that have been used in clinical trials of medicines are by and large asking parents, are things a little better, or are things a little worse, or rate how bad things are right now. And it's really hard to separate developmental changes from a real effect of change. Yeah?
Yeah, just to follow up on that, the clinical global impression, what are your thoughts about that, especially where, I mean, at some level, that's what you'd like to see change if you're a parent or a clinician. On the other level, it's fairly nonspecific, but we don't have a lot of good measures that actually capture change in how somebody's doing. I'm wondering if you could talk about that a little.
So the way I think about, in most studies, the way I think about the clinical global impression scales, and there's one for improvement and there's one for severity, and there's actually now an OASIS scale that essentially is the CGI, clinical global impression, for each domain of difficulty that is seen in most kids with autism spectrum disorder, both core and associated symptoms. If I don't see a change in that scale, it's probably not a medicine that I would prescribe to a kid in the clinic. But if I do see a change, it doesn't necessarily tell me what that means, right? So when I talk about response rate to ADHD medicines, those are all based on the clinical global impressions. It's how much of the time do you see a kid who's rated as much improved? Because if I'm prescribing a medicine for somebody in the clinic and I'm saying like your best case scenario is your child is minimally improved, nobody's going to go for that. So I think that those scales are really important, but they don't really tell us what's changing and why to any extent. And we really need, so when you see a signal there, and I would say for the arbaclofen study, you need another study to understand if there's actually anything there. And there might not be, and I think that's important. But if you really saw nothing across the whole study, then you would say with more confidence that there's nothing there. And I think that this is one of our challenges, especially when we have the heterogeneity. If you see something, you want to know if it's real in a subgroup. But it's really hard to fund that and to prioritize that versus something else. And so in this case, you see some of these big shifts that seem to be driving it. If that's real, I want to know who those kids are. If it's not real, I'd prefer to close the door. And I think that that's going to be an ongoing challenge. I would just note the effects of observation are real, and the Vineland is actually one of the best examples of this. So you ask, and there are all sorts of examples here, but listens to a story for at least 15 minutes. So you ask that at the first visit, and the parent's like, I don't know. Guess who's going home that night and reading their kid a story for 15 minutes, right? So some of these measures that we think are well anchored and concrete will change just because parents listen to what you're asking about and look more carefully to understand what's going on. And then we could also talk about effects of care. So there are real effects of care, and it's really difficult not to care for somebody when they're in a clinical trial, which is to say somebody says, over the last couple of nights he's not sleeping well. Well, has anything changed? Oh, you know what? We've been watching this show together as a family right before bed, and he gets really excited about it. Well, maybe stop watching that show. The next time they come in, he's sleeping much better. Well, that has nothing to do with the medicine. It has to do with being a decent clinician who listens, pays attention, and makes suggestions, even if you're not really supposed to. So, thank you. Any other questions? I'm conscious we might be over time. Yeah, I'm happy to keep talking all day. So,
So you talk about biomarkers in the clinic. I mean, I don't work in the autism field, so I don't have idea. Are there any nationwide efforts to do metabolomics, global metabolomics, in a large number of kids with autism?
So there have been some efforts to do metabolomics, mostly at the level of screening for autism, with the idea being that if you had something that you could do in an 18-month-old, that the pediatrician isn't sure, has concerns, doesn't have concerns, that would help you understand whether to send that child for a full evaluation, that would potentially be quite high yield. There's one of these tests that is just now recently marketed from Stemina. If you look on Spectrum, I think 2 or 3 days ago, you'll see some pretty angry quotes from a number of us about what they're marketing. It does seem like the metabolomics studies highlight subgroups that differ from one another. My read is that overall they're not particularly good at identifying a kid with autism from a kid who doesn't have autism, and in those studies, in most of the studies, instead of looking at kids at the point when you would be thinking of referring them for a diagnosis, you're looking at kids who already have a diagnosis, and usually comparing them with kids where you would never have a concern about autism. That's not the right comparison. There was a large study from Synaptics that then went out of business because of the results, and they haven't published them. I and a couple other people have access to the results, but it takes a lot of effort to put it together. But where they tried to do it the right way, so they did the blood tests at the point when the kid made it to an autism clinic, so they did not yet have a diagnosis, and included the kids who had developmental concerns but not autism. And they did have some signal, but nothing that had a good enough, a robust enough impact to tout it as a potential test. And they didn't try to carry it forward. They said, we don't think that this approach is going to be useful. And they found the same thing for gene expression and for metabolomics. Not published, and so people are still talking about doing this a lot. I don't think it's going to be useful for that. It might be useful for subgrouping. And you can imagine for kids with autism who fit a particular profile that makes you worried about metabolic disease, it's critical to actually do some metabolic testing. But that's quite different than the high throughput shotgun metabolomics that people are doing on the research side. Yeah, certainly. Thank you.
Let's thank Jeremy.